Swayam Prabha. Digital India. Educated India.
So, welcome back to the fourth lecture of the third week. So, in the last class we concluded, or rather we introduced the two molecular players involved in the generation of force, the actin and the myosin. And I even mentioned you that in future we will not follow a gross classification. When you will be teaching here, probably the classifications will go much at the molecular signature. People will ask for, like, tell me the myosin subtype. Depending on the myosin and the actin and what is their kilodalton, what is their size, what is the nature, it is decided, or it can be concluded at least partially, that what kind of force they will generate. Okay. And most of the muscle disorder, like muscular dystrophy or muscle wasting related genetic issues, is nothing but most of the time you will see there may be some form of mutation in these proteins, okay, or related proteins. So, there are two aspects what we will be dealing today. First is, so let us put it together. So, this is where we ended the last class. If you remember, I told you, you have these actins and the myosins which are present there, right. So, today we are starting our week 3 lecture 4. So, let us pick up the story from here where we left in the last class. So, today we will be talking about the two aspects. One aspect what we will be talking about is arrangement of actin and myosin filaments. Second aspect, how this arrangement, so this is your structure, okay. Structure, how this structure leads to the function, and what is the function is force generation. And in the case of muscle, this force generation is also linked to its electrical activity. Now, this is something I am putting a bomb there, like from where this electrical activity came. Okay, I will come to this. Okay, something to do with, I am just putting at this point electrical activity. So, let me digress a little before I come back to this part, the arrangement of the actin and the myosin filament and the structure. So, in our body, most of the tissues, there are multiple ways by which you can classify the tissues. There are multiple techniques based on anatomy, based on anatomical feature, based on function, based on certain other sets of presence of protein or absence of it. But there is another very interesting way you can classify them: electrically excitable and electrically non-excitable. So, in other words, the way you can put it is that the tissues could be classified as electrically excitable excitable and these are non-excitable tissues. So, what does that mean is electrically excitable tissues can generate an electrical waveform what we call as action potentials. Okay? Action potential, which is essentially a electrical waveform due to, or rather generated due to due to movement of ions across the cell. So, I will come later. We are not going to talk about this action potential at this stage, because as soon as we will introduce in the nerve tissue, we will have to come to that. But what I wanted to highlight is, so we have classified these muscles into three categories: cardiac muscle, skeletal muscle, what we are dealing now, and the smooth muscle. Then we went ahead and did another kind of classification in terms of the structural feature. We talk about striated, non-striated muscle, right? That you remember? This is the two anatomical features we talk about, as well as the presence where they are, especially in terms of the location within the body. The third classification which I generated was based on the force generated by them. That was the very beginning. You remember that how I told you that, the cardiac muscle generate a force like this, whereas the skeletal muscle generate huge amount of forces, whereas the force through which the generated in the alimentary canal or the digestive tract, you do not even feel it, right. Now, today I am introducing a fourth classification, but this classification holds true for all the muscles and the nerves are electrically active. Okay. That means, in this context, that means they generate an action potential. It means, in order to generate action potential, they needed to have some specific family of ion channels in them. And the nature or the shape of the waveform, electrical waveform of the action potential of different muscle type is different. So, in other words, this electrical activity could be directly correlated. Mark my word very carefully, this electrical activity could be directly correlated with the force generation of the muscle. So, these two disperse or very separate entities, force generation and electrical activity, could be clubbed together, because we have already talked about the force generation is different for different muscle. Skeletal muscle is K for skeletal muscle, cardiac is CM, and SM is the smooth muscle, right. For these three it is different, and simultaneously, as we will proceed further, you will see their action potential waveforms are also unique. Just keep this in mind, because there are two things now I am telling you to carry over. One, the one which I requested you to carry over is this feature, this structural features. Okay. First thing, and second thing I requested you to carry over is this part. They will come very handy. Just I am introducing them, but we will come back to this. Now, let us continue our story where we left this actin and myosin filaments. Okay. So, now coming back. So, you are having these individual cells or myocytes sitting here like this, which are dividing. This is the nucleus, the second circle I am drawing inside the one. Okay. So, these myocytes are having, I told you, these are the actin and myosin filaments synthesized at a very different, very high concentration. So, initially when these are individual cells, they have a very different kind of arrangement in the individual cells. But then I told you these individual cells come close to each other, right, in the previous class, and then eventually what would happen, they form a tube like structure which is called the myotubes. When they come close to each other, slowly while they are about to lose their individual identity of the cell, these myosin and actin filaments started to rearrange themselves in a very specific geometry. Almost they are aligning themselves like straight lines in the individual cells. Okay. So, let's put it like this. And once the myotube is formed, they come with a very different, they form an arrangement of a different kind. I am just putting it here, and we will come to that molecular arrangement, which was one of the hallmarks of muscle biology. They are arranged in an unique pattern. You see this? You could see a pattern out here in this picture. And what will you observe is very interesting. There are zones, I am just putting where there will be overlaps. Like you see, there are overlaps out here as compared to the places where there are no overlaps. So, it means there are places where the two filaments are like this. They are overlapping, yet there are places they won't be overlapping. There will be overlaps, right? There is no overlap out here, no overlap on this side. Now, if you see it under the microscope, what will you see? This place there is a overlap, right? So, automatically the lesser amount of light will pass through it, because you are seeing from the top, you are seeing from the bottom, it does not matter, right. So, this point there will be lesser light as compared to the light which is following here. So, automatically this side, if you look at this side and this side, the light will be passing more. So, by the simple, from if you are a cameraman, if you are looking at it, this side will look more lighter, this side will look more lighter, this side will look darker. So, what will you observe essentially will be a light and dark pattern once you see it under the microscope. If you see it under the microscope, you will see a light and dark pattern. And these light and dark patterns is what led to conclude that there are some form of striations on them. Those striations, and this was the very first observation. And what people observed was very interesting. This is all back in 1960s, 1950s, we am talking about, even earlier, you know. What they observed, that there is a regular pattern of striations, something like this. And there are, this is nothing but an observation. And then people devoted their life to figure out what is the anatomy of it, and then followed by the, what I told you, what is the anatomy in terms of the arrangement of these actin and myosin filaments, and what is their functional significance. Now, let us see. So, this is how the historically this whole field had moved or is still moving, this first observation. Then people went on to do electron microscopy, and the electron microscopy they could see even much better, because you have the electron density. So, you see more electron dense zones here as compared to the zones which are much more empty. So, still you could see that dense, and as you go further down into this, what you observe was a very interesting feature. And now I am going to draw that feature in front of you. If this is, say for example, my tube I am talking about, what people observe was something like this. First of all, they observe, let me change to go to a very fine thin one, because they observe there are, one second, there are thin and thick filaments which are present there, and there is an unique pattern. I will come more into this pattern. Okay. And on these patterns, people observe something like this. Okay. And in between, what you see is something like this. Okay. And then you observe was something like this. You see a thicker line I am drawing. See? Something, a pattern like this started to emerge. So, the first observation was there are two distinct thin threads which are of protein, which we know, a thin thread and a thick thread. The green one, if you see the picture out here on the slide, these are the thick threads, okay? Whereas you have these threads which are the thin threads. This was the very early observations, and based on that the naming happened, the thick filament and thin filament. Thick filament and thin filament. And if you see this zone very carefully, you will see a lot of overlap out here, right. So, wherever there is a overlapping zone, like something like this, where you see there is enormous amount of overlaps which are taking place. I haven't put the whole red out here, just let me put the red. So, look at it. Now, going back, so these overlaps leads to, the zone of overlap leads to a lot of pattern which is started generated, and this whole zone is sometime in the literature they call it is the A band, where I have put it, and there is a zone where you will not find these blue lines, that is called, that is a very light zone which is called the I band. I do not get bogged down into this I and Z band, but you just try to understand. Now, there are a lot of variations, and this is called the Z lines. Okay. And of course, I have marked the thin and thick filaments, and apart from it, these thin and thick filaments are, these thick filaments are attached by a unique kind of, just let me pick up another color which will kind of, there are attachments which are there like this, and those attachment molecules which are present there are called titin. So, I introduce thick filament, thin filaments, and the Z lines and the A bands, right. Now, what are those thin and the thick filaments? Now, these thick filaments are myosin proteins, and these thin filaments are actin proteins. Okay. Now, going back to this structure, now see, So, here you are having, so how we marked it? Okay, myosin we are calling as a red. Okay. So, these are your myosin and these are your actin. Of course, we have just changed the color code here. We have the actins shown in red here and the myosin shown in blue, but that does not matter. So, this is how the architecture started to emerge here. And if you go a little bit further, if we draw it a little bit in a much more finer way, it will be something like this. Thin filament, thick filaments first. So, the thick filaments are like this, sorry. Okay. So, this is how the thick filaments look like. Now, I am kind of at a very, very microscopic level. These are the thick, one second. Okay. These are the thick filaments, and within the thick filaments, you will come across some very interesting feature. I will come to that. What are those features you will come across? Within the thick filaments, you will see something like this. We will come to that, what are the significance of those structures. Okay. So, this is about the thick filaments. Now, I will go to the thin filaments which are present there. So, thin filaments are in close proximity. Let us take a simple scenario out here. Or another one out here, another one out here, and maybe another one like this. Okay. So, I am just keeping it okay. Okay. These are the Z lines. You remember in the previous slide I talked to you about the Z lines, just for those, just recap. So, these are the Z lines. Okay. So, now, and these thin filament, thick filaments are attached to even much more thinner, which are called the a spring like structure, which are called the titin molecules. Okay. Now, here is the architecture. Right. Now, 1 minute, I have not finished it off yet because I forgot to put, right. Now, I have the structure ready for you. So, this is how the structure looks like, as if they are attached in spring. Now, this structure is actually move like this. In other words, this framework what you observe out here, this framework, within this framework you see this spring. So, there is a movement of these thick filaments on the rails of thin filaments. Again, carefully listen to me. So, there is a movement of these thick filaments on the rails of these thin filaments. Translating it in terms of the molecules, there is a motion of these myosin filaments on the rails of the thin actin filaments. How that get executed? Okay. Once again, another repeat. There is a movement of, there is a controlled movement of the myosin thick filaments or the myosin filaments within bracket the thick filaments on the rails of the actin filaments, which are thin filaments as shown here on the red on the slide. Okay. Now, there are few terminologies which needed to be defined. I defined this terminology, the Z line. Okay. We know about this titin molecule. We have talked about myosin. Within myosin, there are few other interesting things. We have talked about this part. This is called H zone. Okay, this I am just introducing. I haven't introduced this earlier. And this whole assembly from this end to this end is, there is a name for this assembly. This is called sarcomere. Okay. This one single unit is called sarcomere. Then this myosin, this is called M line. Okay. And this I have already talked about, the Z line, and these are the myofibrils. Myofibrils. And the movement out here is something like this. There is a movement like this. Okay. Contraction and filament sliding. There are two words now I am introducing: contraction and filament sliding, which is also known as sliding filament theory. And I have already explained it, what is sliding filament theory. Sliding filament is nothing but, which I repeated, on the rails of the thin actin filament, the myosin slides like this. But is this process a spontaneous process? If it is a spontaneous process, our muscles will spontaneously will twitch. No, it is not a spontaneous process. There are certain muscles where it is a spontaneous process, but it is governed by lot of neural signatures and the muscle signatures. Now, this is where the physical movement of the muscle occurs. Now, if you imagine the slide which I bring, so imagine at every level if this kind of movement are taking place, it has to be orchestrated. So, there has to be some kind of a control which is governing them. The one which I am drawing now is basically a nervous control which I am introducing. Okay. At this point, there are few other molecular details which I will be going out here, this zone, which is called as the myosin head. So, let us close in today, and the next class we will go to the further molecular details of this structure, and then we will talk about how this sliding filament theory gets executed and how much force is generated. Thank you.